So we're here for the second Keystone meeting on phenotypic drug discovery. Drug discovery was historically carried out using a phenotypic approach. That all changed in the early 1980s and 1990s when we switched to more of a target-based drug discovery approach. That has had its successes with well-validated targets. However, what we've realized for the past 10 years is that phenotypic approaches still have their place. They can enable us to discover novel biology and first-in-class therapeutics. What we need to be conscious of, though, is that what we think of in terms of phenotypic screening nowadays, which is cell-based assays with tens of thousands of compounds being tested, is not actually how most drugs were discovered using phenotypic approaches. A lot of the time, we've used complex animal models or even human data to understand a link between a compound and a disease indication. A lot of these discoveries were not straightforward either. They were not planned. They happened through serendipity. So we need to take that into account and moving forward, develop strategies, best practices in order to prospectively deliver this novel biology and these novel first-in-class drugs. And that's the goal of this second Keystone meeting.
And we are seeing now that the approach is delivering drugs. This is based on the well-defined endpoints and appropriate assays that are driving these success stories, which are new drugs for schizophrenia, cystic fibrosis, anti-malarial drugs. So we are now seeing the results of the resurgence of this approach in these new drugs that are coming and will be invaluable for the patients.
Mm-hmm. We learned a lot, I think, from the participants at this meeting about the current state of phenotypic drug discovery. What in your mind are some of the most important observations that have been made or advances that have been made?
Well, maybe in my mind is that phenotypic approaches can uniquely identify some specific mechanisms of actions that would be difficult to identify for target-based programs, such as drugs having polypharmacology. I'm thinking about the Roche clinical candidate for hepatitis B, for example.
Right, I agree. I think another thing we've learned is that the field isn't settled yet on the question of whether or not we need to have a target in mind for a phenotypically discovered drug before that molecule advances into clinical development or into regulatory consideration.
But I think that was a very heated debate the first meeting, and we were all jumping over each other trying to say it's become much more mature, the discussion, the debate, and it's really sort of centering itself around, is it because it's a need to de-risk? Is it because you need it? Some indications are better suited to not having the target at the moment.
I agree. I think that's been something that's come out, and that nuance is becoming clearer and clearer on a case-by-case basis.
The other thing is, I think pharma, surprisingly, is much more on board. That's the other thing. It seems like the first meeting was a lot about, do we need phenotypic drug discovery?
Right.
And this one started off by the premise, yes, it's something that's good, and like you said, it started to deliver candidates. It started to deliver drugs. We're starting to see the first successes.
One of the things I thought that was interesting is that the insights from the biology might advance our ability to identify the targets.
Yes, and these discussions about target identification, the risking, profiling, this has been in a more advanced level than previous discussions. I think now, even if we don't identify the target, profiling or fingerprinting of these compounds is essential. So we may then go and find a target, and maybe that will help the optimization process, the clinical biomarker identification. But still, so we are moving forward with these discussions, and I think this is very beneficial.
The other thing that has become sort of cemented in this meeting, I think, and everybody agreed on, is the need for the assays to have well-defined endpoints, to have them be very close to the disease, the mechanism in a dish that keeps popping up and up. And
I think another thing that has emerged from this meeting in particular is the ability of phenotypic drug discovery to give rise to completely unanticipated molecular mechanisms of action.
Yes, and I think part of the reason why pharmaceutical industry is becoming more comfortable with phenotypic drug discovery is because some best practices are emerging.
Yep.
In terms of assay design, but also hit triage, target identification, safety de-risking. So with new strategies, best practices, people feel that they have the data that is sufficient, that is necessary to move forward to the clinic.
And that came up several times during the meeting.
Yeah.
And so I also wanted to point out new ideas about polypharmacology, smaller molecules that can be more suited for the phenotypic approach with lower potencies, and with these pharmacology approaches maybe helping have less safety concerns. So particularly these new concepts, I think will be unique to phenotypic drug discovery and very useful to move forward.
Definite consensus on that. There's certain mechanisms of actions that we would have never been able to find if it hasn't been a phenotypic drug discovery. And that was seen in Lipinski's talk, and who was the other? Laura Kiesling. These things, even today, Ben's talk about how the stoichiometry was so low. Had it not been a phenotypic screen, never been able to pick it up.
That was very interesting, yes.
So I think that's another thing that sort of there's consensus around now.
Yes, and I think, so we've also learned a lot about where the field is heading and the types of the new technologies that are driving the field forward at a tremendous pace. Artificial intelligence is one in terms of analyzing the assays. I think also the sophistication of the phenotypic of the models that are used in phenotypic screening themselves,
and also reaching out to real-world data, omics data from large-scale databases. This was Clemens Serser's talk, which was very interesting, linking from phenotype to patient, clinical phenotype, and making sense of the whole story. That was really something very innovative for us.
There were remarkable models that used primary cell data. There was the skeletal myoblast talk. I think we had a fantastic talk on zebrafish and looking at behavior. That was wonderful. There were a number of there was a couple of other primary cell models discussed.
Even looking at patient data.
Yes.
Some of the models that are being, or assays that are being done on patient data. I think the rare diseases, the monogenic diseases, and being so close to the patient data has been transformative.
And what I noticed as well is the use of in vivo model, especially for CNS disorders.
Yes.
Because these are diseases that, frankly, we cannot recapitulate correctly in a dish.
Yes.
So the use of animals, I think, was very useful.
Right.
I think this also came up in the 2016 meeting as a new thing, which looked a bit ambitious, but right now it's like, again, like we said before, kind of a standardized approach that is really, again, delivering new candidates.
To that point, polypharmacology was kind of like a thought in the 2016.
Yes.
Now we're talking about
Yes.
how are we going to use it properly? How are we going to take advantage of it?
Which is interesting, because it used to be that polypharmacology was seen as a disadvantage of phenotypic screening. And in fact, it turns out that it's an advantage. I mean, you remember the Roche story? Inhibiting two enzymes. But if you inhibit just either one, that's not enough. Right. You know, the CNS compounds, you need to modulate multiple receptors.
Cancer compounds as well.
Yes.
And we wouldn't see this without the complexity of the phenotypic assays. And the scale of these assays is also, and the sophistication, as I said before, is improving. There were a number of talks on iPSC-based models, and they had sophistication we've not seen previously.
They also make for really cool slides with beating hearts and things like that.
Yes, I'm very partial personally to beating hearts. Yes.
True.
I think one way in which we can get the most value out of phenotypic approaches is by coupling them with target-based approaches. The talk from Chanwa about the bed domain inhibitors, going from a pan-bed domain inhibitor to selective bed domain inhibitors, where you can target different proteins, different domains, and then you have different pharmacology. That represents quite an opportunity.
I like that she not only called it complementary, but synergistic. And it's not pitting the two methodologies one to the other.
Exactly. And that's also something that I think has been unique to this meeting, this integration between the target-based and phenotypic-based approaches. So we are talking more now about drug discovery as a whole, and then we can do target or phenotypic depending on the project or the phase of the project. So this is also good that it's been integrated by the community. We are not longer ashamed of doing target or phenotypic screening.
So along the lines of what you said, so you've raised a very good value-added concept here of the target plus the phenotypic screening. Another value-added benefit that you alluded to was the interactions between industry and academia. I think that's an important part of this meeting, that we have such a large, for Keystone meetings in particular, to have such a large pharmaceutical company presence at the meetings. I would say probably half of the people, so we're from pharma companies, and about half of the people from academia. And it was phenomenal interactions. And I think that's leading to a lot of collaborations being set up. And that started at the last meeting, and I think that has continued at this meeting. You've gone over to one side to the other. Yes.
You represent the transition.
I'm the transition.
That's right. And to add maybe even a little bit more granularity to the industry part of the audience, it falls in biotech and big pharmaceutical company buckets. And these two entities have different approaches to phenotypic drug discovery. And I think some of the discussions have been robust on the concept of target ID, safety de-risking, clinical progression without a target. I think these discussions make each side richer and maybe more aware of what needs to be done or the possibilities.
That's right. And I think through the collaboration, because of the different cultures on these two sides and the different ways that projects are decided upon, funded, et cetera, I think the collaborations can be particularly powerful.
I think the panel discussions were very productive.
In that regard.
I felt very comfortable during the interaction, being part of one of the discussions. So that was really good.
Yes, and there were some good discussions. I mean, I remember the safety de-risking panel. We kind of came to a consensus that while the FDA requires two animal toxicology studies in two different species, we would want to run these species with a proof of pharmacology for each of the species. So even if we don't know the target, at least we know that we are engaging the mechanism, so that we get a chance to see whether there's a safety liability in these in vivo models. So
that was one big thing, the difference between target ID and mechanism and how they're complementary.
That's very important, because for some people it wasn't clear, I think, until very recently. And during these discussions, it may have become more clear for them.
Right. And that extends to when you think about mechanism, not just a molecular mechanism for the protein, but also a biological mechanism of action that these assays might capture. And that could include interactions with many other proteins. And in that regard, it was intriguing that so many of the compounds that people have talked about have altered the ability of a protein to interact with another protein. So that's a fairly novel mechanism of interaction. I mean, mechanism of action.
And something that will be, frankly, difficult to get to using a target-based approach.
Exactly.
These are difficult to predict. So, with a phenotypic screen, you just discover it.
Right.
I think one of the things that is becoming clear is that there's going to be a big need for data wrangling, these profiling methodologies, all of these things that are coming together. And we're seeing the beginnings. So I think that's the opportunity, but also the challenge, how to do it and how to do it right. Somebody said that the hype around AI, so just make sure that we get it right. I think that's one of the big challenges.
Well, for me, it was illuminating how you can divide this problem into different levels of complexity. So a first level of complexity is how do you drive the, say, advancement hit to lead of a compound from a multi-parametric assay. So you might have multiple parameters to look at. How do you integrate that information? And then there are levels where you look at phenotypic assays where you can have many, many, many parameters that you don't even understand. And the question is then which ones to pay attention to, how to. And I think that's where the computational tools really come into
these
their own.
Tools, they help us. You may not understand the full picture, but you can compare with other compounds. And based on the properties of those other compounds, then you can draw conclusions which may be useful for your partially unknown.
So the increasing use of image-based, high-content screening, either static images or we and others were describing single cell or kinetic imaging where you're acquiring images at hundreds of frames a second. I mean, there's a phenomenal amount of information that's untapped, and I think that's going to be a huge opportunity in the future.
Even non-biased imaging, which in the phenotypic spirit, it's really, I think it can be very useful.
Yes.
I think one of the themes that's going to be the hardest to wrangle, it's a little bit of what Derek said in his talk, but this finding the right assay to quickly go into translation. I like the phrase, make the chain of translatability tighter. I thought that was, to me, that was kind of one of the things, and I think it's going to be a challenge for the field moving forward.
I think that's the fundamental issue here, is bringing the patient back, the patient context, back into these early stages. And so bringing the endpoints of our assays that are in vitro so to as close as possible to mimic the clinical presentation.
Exactly.
But the clinical presentation is a patient in front of you, and the assay is some readout in a dish that may be image-based. And the question is, you don't know how that image relates to the patient. And I think that's where the reference compounds and the use of artificial intelligence will have a huge impact in the future.
And something else to consider is that we might do well to reconsider the use of in vivo animal models.
Yep.
I mean, these have gone out of fashion for screening. But when you look at the progress that's being done in the CNS field, this represents a good opportunity. I would argue that for the diseases that are maybe multi-organ diseases, or that we don't understand very well, if there is an animal model that is highly validated, that may be the way to go. And what we can do is partner that model with a subset of compounds. We heard that from Chris Lipinski and from the Blue Oak pharmaceutical scientist. You can take molecules that are very small, fragment-like, and then dose them in an in vivo model, and you have a surprisingly high hit rate.
Right.
So there are opportunities there.
Yes. And we heard an interesting talk at the beginning from Maurizio Palecchia using fragments on molecules so that you might be able to go from fragments to small molecules that work with more traditional pharmacology. And so I think that this is also a very interesting area.
And related to what you said, libraries are getting smaller because we understand better what we want from this screen. So this may be converging with the fact that we can use in vivo models to do phenotypic screening directly.
Yes, we should use smaller compound libraries. I mean, libraries, not smaller compound libraries.
Libraries of smaller
compounds.
Both. I would say both.
Yes. In both in vivo assays as well as in vitro.
Yes, we want larger, more complex libraries of smaller compounds.
Smaller libraries with smaller compounds. That's the
with
thing.
pharmacophores that we like.
Yes. So I see one more area for opportunities as well as challenges. And for me, that is safety de-risking and translation as well. Basically, it's about what is the information that we can acquire short of getting a target identified that can enable us to move forward into the clinic. And so I can think of some complex in vitro safety assays that could allow us to identify sooner some toxicity signatures, thinking about the biomap assay panel, zebrafish, for example.
That's right.
And for those projection into the clinic or to make sure we cover C efficacious in our toxicology study, we need to identify biomarkers or signs of pharmacology, so to make sure that we are covering that C efficacious in animals and that we will cover it in humans.
And we can couple those assays, those models with transcriptomics, omics in general, so that we understand better, even if we don't know the target, again. But this will make people feel more comfortable about their project as it moves forward to clinical phase. True. I wanted also to raise the point of the younger people attending the meeting.
Yes.
I mean, they are learning phenotypic screening now as they enter the scientific world, and I think this is an advantage. So it took, I would say, a generation of scientists, so it's taken a generation of scientists like us, like me, for example, who I used to work in target-based alone. But these young people is seen, or is this more mature phenotypic screening area. And I think this is going to be very beneficial.
Because Chris Lipinski this morning was saying, I only did, I'm like, really? It's like 10 years I did nothing but phenotypic. And then sort of this whole debate.
Towards the end of his career.
Yes, and he was
Yes.
saying that, what was it, that he's a valuable yet exceedingly rare
resource.
We have to take advantage of this generation before.
And then a lot of young people, Christy, who gave the poster, the last talk this session, what is his name? He's asking all the questions, very engaged in the meeting, very taking part in it, asking questions.
Yes. There is a talk that intrigued me from Hassan Al-Ali, University of Florida, and it was about the prospective use of polypharmacology, so that we can run a phenotypic screen and accidentally discover a drug that will act in that fashion. But I thought it was quite clever to try and preempt that and design polypharmacology into the assay. And his example with neural outgrowth was quite interesting.
Yes.
So I'm looking forward to seeing that evolve over the coming years.
And that reminded also is similar to Ben Cravat's discussion of the activity map. So being able to characterize proteins in the cell by structural means, but also relate that to their activity in the cell by phenotypic means.
And I think also this talk from Scott Walker, I think about antibacterials, where you could sensitize the bacteria or partially mutate the bacteria so that they would be more sensitive to a second mutation. And so this is also related to polypharmacology. You'd be looking for 2 targets at least, and opening up to new mechanisms of action that would go unnoticed in a single attempt on in a single mutated bacteria.
Phenotypic drug discovery entails a collaboration between chemists and biologists. And so what I've learned is that when chemists question the throughput of an assay, I can safely ignore them. However, when they start questioning the disease relevance of the assay, that's when you have to pay attention. That's when you have to listen. This is why I was listening carefully when Derek Lowe presented on the use of predictive validity for the development of our assays. He utilized the paper from Jack Schenone and Jim Bosley, a great paper that highlights that there is really a lot of value in using more predictive assays. In fact, there is a figure that suggests that testing 100 compounds in a highly disease-relevant assays will be more productive than testing in 1 million in a poorly predictive assay. And this is important because at the end of the day, regardless of the sophistication of our approaches, of our tools, or how clever we are in our approaches for characterizing the hits, if the original assay was not disease-relevant, the project is very unlikely to deliver anything of value. So this is, I would suggest, the most fundamental characteristic of the field. We need to develop highly disease-relevant assays if we want to be successful. If the assay is not disease-relevant, the project will have no value.
Right. So in fact, we should be quite critical as a group or as a field on triaging assay thoughts and assay concepts before they get to the point of even being developed. If the assay seems not to mimic the clinical presentation that you're trying to address, I think that we shouldn't go forward with it. I think what we should do is have quality, some form of quality control that we can look at the assay and judge, is this assay going to return the compounds that we think are going to ultimately be effective?
Back to the
Again,
tightness of the translatability.
The tightness of the translatability.
And I think that's where phenotypic drug discovery is going to, the hinge for it,
right.
I think.
And there were a lot of discussions about returning to that throughout this meeting. And so I think that's an emerging awareness that is going to become stronger as time goes on.
I think for those of us practicing phenotypic screening with in vitro models, which may be based in patient cells, in diseased patient cells, you realize that that may not even be enough. You need to have more complexity. You need to have the immune system cells there.
That's right.
To have the whole dynamic situation that you can address with the phenotypic screening. So maybe 3, 4, 5 years ago, we used to talk about having patient cells as very relevant or relevant enough, but now we're thinking in leaping into the next level of getting as close as possible to the real patient.
So I think this highlights then something for the future, and that is to recruit to the field people who don't think of themselves as involved in drug discovery, but nonetheless have particular expertise in recapitulating critical aspects of a disease presentation. And I think bringing those people to the field is a task that, as organizers of this meeting, that we should encourage.